Why does every gardener, in every country, on every continent, dig up the same small clay-colored spheres from beneath their soil, and then immediately throw them away, convinced they are pests, eggs, or something diseased, when in fact they may be the single most important structure your garden has ever produced? You have almost certainly held one. You may have crushed it between your fingers without thinking. And the strangest part of this story is not what these tiny pots are. It is who built them. And why nobody told you. Long before there were gardening books, there were warnings. In rural Japan, farmers spoke of tsuchi no tsubo, the earth's little jars, and were taught never to disturb them when turning a field. In parts of pre-industrial England, hedge-laying manuals from the 18th century mention small, hollow clay beads found tangled in old root systems and instruct laborers to leave them be. Indigenous agricultural traditions in the Andes, in West Africa, and along the Mississippi all reference, in different words, the same quiet rule: do not remove the small pots. For most of human history, people who lived close to the soil understood that something in the dirt was making vessels, perfectly round, hollow, often arranged in clusters, and that those vessels mattered. The knowledge was practical. Then, somewhere between the rise of synthetic fertilizer and the invention of the rototiller, that knowledge quietly died. For nearly 200 years, soil biologists have been recording these tiny structures in their field notes. They have appeared in core samples from temperate forests, in archaeological digs in Mesoamerica, in pasture studies in New Zealand. Journals from the 1880s describe them. Mid-20th century mycology textbooks illustrate them. They were never secret. But they were never explained to the public either. The agricultural extension pamphlets that reached your grandparents reduced the entire underground world to 3 letters: N, P, and K, nitrogen, phosphorus, potassium. Everything else, every spiral, every chamber, every tiny clay pot tucked between the roots, was treated as background noise, irrelevant, invisible. And so, generation by generation, we forgot how to see them. Even the scientists studying soil openly admit that we still do not fully understand what is happening beneath our feet. A single teaspoon of healthy garden soil contains more individual organisms than there are humans on Earth. And the vast majority of them have never been named, let alone studied. Soil ecology, as a serious interdisciplinary field, is younger than most people watching this video. The first comprehensive genetic surveys of subsurface microbial communities only began in the early 2000s. The picture we have now is, by every honest admission, a sketch, which is why what follows is offered carefully. Not as conclusion, as unveiling. The first thing to know is what those tiny pots actually are. They are not eggs. They are not seeds. They are not the cocoons of any insect you have been warned about. The small spheres you find when you dig, usually between 2 and 8 millimeters across, often a pale tan or rusty brown, sometimes clustered like tiny grapes, sometimes scattered like beads, are aggregates, mineral organic aggregates, to use the formal term. They are tiny architectural structures built by soil organisms working together over months, sometimes years. The walls are clay, bound together by polysaccharide glues secreted by bacteria and fungi. Inside each one is a microscopic city. But here is what the old textbooks missed. Until the late 1990s, these aggregates were treated as passive, accidental clumps of dirt, mechanically stuck together by moisture and root pressure. Then, in 1996, soil scientists using new staining techniques noticed something strange. The clay particles were not randomly stuck. They were oriented, arranged, layered in patterns that suggested deliberate construction. The pots, it turned out, were being built. And then, in 2004, a research team studying earthworm castings realized that worms were not merely producing waste. They were producing engineered structures, lining the inner walls with secreted mucus and seeding them with specific bacterial strains carried in their gut. Each worm was, in effect, a tiny ceramicist, firing little kilns of microbial life and leaving them behind in the soil. And then it got stranger. Because earthworms are not the only builders. Mycorrhizal fungi, the thread-like networks that wrap themselves around plant roots, also produce these aggregates, gluing soil particles with a protein called glomalin. Glomalin was only formally identified in 1996 by a USDA soil scientist named Sarah Wright. Before that, nobody knew it existed. We now estimate that glomalin may account for up to a third of the carbon stored in some soils. A third of the carbon, stored by a substance we did not know was real, produced inside structures we were taught to ignore. Inside each tiny pot, the conditions are different from the surrounding soil. The oxygen levels are lower. The moisture is held more steadily. The microbial population is denser and more diverse. They are, functionally, microscopic refuges, pockets where soil life can survive drought, flooding, freezing, and chemical disturbance. When the surface soil is scorched by sun or stripped by rain, the inhabitants of the pots wait, and when conditions return, they emerge and they rebuild the world above them. This is why traditional farmers told you not to disturb them. I'll be honest, this one shook me. I had to sit with it for days. I have spent years pulling weeds, turning beds, breaking up clods of soil with the side of a trowel, and I have, without knowing, destroyed thousands of these structures, crushed entire microscopic civilizations between my fingers because I thought I was preparing the ground. The thought is uncomfortable, but it is also, I think, the beginning of something better. Because once you know, you cannot unknow. And the next time you kneel in the dirt, your hands move differently. There is more. The pots are not only refuges, they are also batteries. Soil aggregates of this kind hold water at a tension that plant roots can actually access, meaning a garden with abundant aggregates can survive droughts that would kill the same garden in the same soil without them. Studies from agricultural research stations in Iowa, in Kenya, and in the Australian wheat belt have all shown the same pattern. Aggregate-rich soils need significantly less irrigation. In some trials, up to 40% less. They are, quite literally, the reason some farms survive a dry year while neighboring farms do not. And then there is the carbon question. Soil scientists now estimate that the world's soils contain roughly 3 times as much carbon as the entire atmosphere. The majority of that carbon is not loose. It is locked inside aggregates, inside these very pots, where it is protected from the microbes that would otherwise digest it and release it as carbon dioxide. When we till, when we compact, when we strip vegetation, we shatter the pots, and the carbon comes out. A significant fraction of the carbon emissions attributed to agriculture worldwide is not from machinery or fertilizer. It is from broken aggregates exhaling, for the first time in decades, what they had been quietly holding. But here is what nobody told you. The pots are also where antibiotics come from. Most of the antibiotics we have ever discovered, penicillin's relatives, streptomycin, tetracycline, vancomycin, were produced by soil microorganisms living in exactly these aggregate microhabitats. The reason aggregates produce so many antimicrobial compounds is the same reason cities produce so many laws. When organisms live in close quarters, they evolve elaborate ways to negotiate, defend, and signal. Every tiny pot in your garden is, in a quiet way, a pharmaceutical laboratory that has been running for longer than humans have grown food. And the most recent unveiling, from research published in the last few years, is perhaps the strangest of all. Soil aggregates appear to communicate. The fungal threads that wrap around them link one pot to the next, and to the next, across distances of meters. Through these threads, the microbial communities inside the pots exchange chemical signals, warnings about drought, alerts about pathogens, even nutrients passed from one plant to another through the network. Your garden, beneath the surface, is conversing, and the tiny pots are its libraries. So the question we started with, why does every culture, on every continent, tell you to leave the small pots alone, has an answer that is partly scientific and partly something else, something older. The people who lived closest to the soil knew, without instruments, that the dirt was doing work, that the small round things were not debris, that to break them was to lose something you could not see and could not easily replace. And here is what I cannot resolve. I do not know how they knew. I do not know whether it was observation passed down through generations, or whether the human eye is quietly capable of recognizing patterns the mind cannot articulate. I want to hear your theory. Drop it below. The strangest one gets pinned. Tell me what you were taught about the soil where you grew up. Tell me what your grandmother said, or what your grandfather did, or what the old farmer next door warned you never to disturb. We are reconstructing a knowledge that we almost lost because the next question we'll ask is even older, and the answer was hiding in your grandmother's garden the whole time.